What if I told you that your body can identify and destroy pathogens without producing antibodies, but simply by recognizing the sugar on the surface of the pathogen? That is how the Mannose-Binding Lectin pathway of the complement system works. A quick recap: what is a complement system? I've covered that in a previous video. You can do well to check it out. The complement system is a group of glycoproteins that work together to enhance the activity of the phagocytic cells and also antibodies. So from the name complement, you know it means to enhance. Now, this thing is achieved through three major pathways. We have the classical pathway, we have the lectin pathway, and then we have the alternative pathway. I have covered the classical pathway in its separate video, so you can check that out. But for the lectin pathway, which is what we're going to be looking at in this video, it involves the recognition of sugar on the surface of the bacterial cell. So that is how our body can be able to destroy pathogens without necessarily waiting for the production of antibodies. Now, this pathway, the Mannose-Binding Lectin pathway, simply from the name, you know it means that there's a binding of mannose happening somewhere, right? So this protein, this lectin, recognizes mannose on the surface of a bacterial cell and binds to it. We all produce Mannose-Binding Lectin. Our body produces it. So what it does, that it patrols the blood looking for mannose. The moment it recognizes mannose on the surface of any bacterial cell, it binds immediately to it. Now, when it binds to it, this is the activation of the lectin pathway. Immediately it binds to it, it activates these proteins that accompanies this protein, the MASP-1 and 2. The binding of this lectin to the mannose activates this protease. Now, the function of this protease is to cut. Immediately it activates this protease, C4 is cleaved into C4A and C4B. C2 is cleaved into C2A and C2B. Now, remember we have said that in the complement system, the larger fragment is B and the smaller fragment is A. Exception of the C2 protein, in which the larger fragment is C2A and the smaller fragment is C2B. So after this protease, the MASP has cleaved C4 and C42 into A and B. This smaller size, the C4A and the C2B, diffuses out, while the C4B, it now becomes C4B and C2A. This becomes the C3 convertase that cleaves C3. The formation of the C3 convertase leads to the cleaving of C3 into C3A and C3B. At this point, the pathway follows a similar pattern with the classical pathway. Remember, we've said that at some point, the outcome for these pathways is the formation of the membrane attack complex. So once C3 has been cleaved into C3A and C3B, follows a similar pathway as a classical pathway. The C3A goes ahead to become an anaphylatoxin, a powerful inflammatory molecule, right? The C3B serves two functions. The first one is to opsonize. It coats the pathogen to enhance inflammation, and then it goes ahead to bind to this, the C4B, the C2A and the C3B, to continue the reaction. Now this becomes the C5 convertase. Now this C5 convertase recruits C5, cleaves it into C5A and C5B. C5A moves into the serum to become a strong anaphylatoxin and inflammatory molecule, while C5B initiates the formation of the membrane attack complex. So from there it goes C5, C6, C7, C8, C9 up to the formation of the membrane attack complex. So for the Mannose-Binding Lectin pathway, the final thing you should have is the C4b, the C2a, the C3b, C5b, C6b, C7b, C8b, and C9b. This is called the membrane attack complex. The function is to puncture holes in the cell membrane of the pathogen and lead to the lysis of the cell. So you just understand the principle. The Mannose-Binding Lectin binds to mannose on the surface of the bacterial cell, which activates the mannose-associated serine protease that cleaves C4 and C2 into C4a and B, C2a and B, which leads to the production of the C3 convertase. And at that point, all the pathways converges. First way to understand the lectin pathway: sugar binding pathway. Mannose binds to mannose, activates MAPs that cleave C4 and C2b. So that's the simplest way. All of them leads to the formation of the membrane attack complex. So when you're writing it, you are not just writing B, 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 B. You understand what you're actually writing. So in another video, we are going to look at the final pathway, which is the alternative pathway.